Death is rarely a single, instantaneous event. It is a predictable, progressive, thermodynamic cascade. Modern medicine distinguishes between clinical death, the cessation of heartbeat and respiration, and biological death, the irreversible failure of cellular architecture. Clinical death is a reversible state. If oxygen delivery is restored quickly enough, the patient can be resuscitated and organ function preserved. The bridge between temporary clinical arrest and permanent biological death is defined by a single mechanical failure, systemic hypoxia. This lack of oxygen initiates the sequence of cellular and systemic collapse. The transition from life to death is a failure of homeostatic oxygen delivery, rendering the biological system unsustainable. Even as individual organs shut down at different rates, the brain dictates the point of no return for human life. This section illustrates brain death, defined legally and physiologically as the irreversible loss of all brain and brainstem function. Diagnostic criteria for this state require zero consciousness, the absence of spontaneous breathing, and no brainstem reflexes, even when stimulated by carbon dioxide. Brain neurons are uniquely vulnerable to hypoxia because they have virtually zero glycogen stores, relying entirely on continuous oxidative phosphorylation to survive. Without a constant supply of oxygen and glucose, irreversible hypoxic injury begins in these neurons within 3 to 5 minutes. A patient in this state can be mechanically maintained, with a ventilator and drugs sustaining a heartbeat, but this artificial circulation masks the loss of biological viability. Because the brain is the body's central integrating processor, its permanent failure guarantees systemic collapse, which is why brain death equates to legal death in most jurisdictions. When circulation stops, the focus shifts from systemic organs to the interior of a single cell. As blood flow stagnates, oxygen delivery drops to zero. The immediate biochemical consequence is the shutdown of oxidative phosphorylation within the mitochondria. This diagram shows the resulting ATP depletion. Without oxygen, the cell cannot produce adenosine triphosphate, the molecule required to power its basic functions. In response, the cell switches to anaerobic metabolism, a temporary measure to generate trace amounts of energy without oxygen. This emergency pathway produces an excess of intracellular lactic acid. The drop in pH creates severe intracellular acidosis, which begins to physically denature protein structures and fragment DNA. ATP acts as the currency of biological order. Without it, the cell is forced to dismantle its own structural architecture. The most immediate casualty of ATP depletion is the sodium-potassium pump, a mechanism that requires constant energy to maintain the cell's electrochemical gradient. As the pump fails, sodium and water rush across the membrane, causing the cell to undergo extreme swelling. This swelling is followed by a massive influx of intracellular calcium. High concentrations of free calcium inappropriately activate a cocktail of digestive enzymes, including phospholipases, proteases, and endonucleases. These enzymes act as microscopic scissors, physically shredding the cell's internal lipid membranes and destroying organelles. Once these enzymes breach the mitochondrial membrane, the cell passes the threshold of irreversibility. It cannot recover even if oxygen is reintroduced. Biological death involves this active, catastrophic process where the cell digests its own protective borders. This chaotic breakdown sits in direct contrast to the body's method for healthy programmed cell turnover, known as apoptosis. Apoptosis is a tightly regulated, ATP-dependent process that shrinks the cell and packages its contents into neat vesicles without triggering an immune response. Because post-ischemic cells lack ATP, they cannot execute apoptosis. They are forced into a chaotic state of necrosis. Under extreme stress, cells will also trigger specific regulated necrotic pathways, such as necroptosis and pyroptosis. In pyroptosis, the activation of the enzyme caspase-1 leads directly to the cleavage of a specific protein called gasdermin D. The cleaved fragments of gasdermin D then oligomerize, forming large lytic pores directly in the plasma membrane, which ensures violent cell rupture and systemic inflammation. When these membrane-rupturing protocols execute across billions of cells simultaneously, the micro-level structural failure scales up, causing irreversible organ collapse. These microscopic cellular failures manifest as the macroscopic, visible changes observed in forensic pathology. Once the heart stops pumping, gravity takes over. Blood pools into dependent areas of the body, creating the physical markers known as pallor mortis and livor mortis. As metabolic heat production ceases, the body undergoes algor mortis, thermodynamically equilibrating with the ambient room temperature. The ATP depletion observed at the cellular level is also directly responsible for rigor mortis. Muscle fibers rely on ATP to release the bond between contractile proteins. Without it, myosin heads lock permanently onto actin filaments, turning muscles rigid. This rigid state typically sets in 1 to 3 hours after death, peaking between 12 and 36 hours. These forensic hallmarks are direct readouts of the body's shifting thermodynamic state as cellular energy production ceases. Following rigidity, the body enters the active decomposition phase, starting with autolysis. The intracellular acidosis built up during the early stages eventually dissolves lysosome membranes. These organelles burst, spilling digestive enzymes and causing the tissues to liquefy. Autolysis clears the way for putrefaction, a process driven primarily by the body's own endogenous bacteria. Without an active immune system to keep them in check, intestinal bacteria switch from symbiotic digestive helpers into consumers of surrounding tissue. As these bacteria digest the host from the inside out, they produce quantities of gas that lead to bloating, discoloration, and skin slippage. Forensic pathologists rely on the overlap of these stages, algor, livor, rigor mortis, and putrefaction, to calculate the post-mortem interval and determine a precise time of death. Death operates by strict thermodynamic rules. When the continuous input of oxygen and energy fails, the architecture of the human body is disassembled, step by step, until it reaches thermodynamic equilibrium with its environment.